Hi, this is Dr. Josh with Alzheimer's Breakthrough, and I just had the pleasure of interviewing DrBeen. Amazing interview talking about not just the spike protein, but how it may interact with Alzheimer's disease and other brain pathologies, diseases. His explanations were right on. He knows the latest papers in this field and was willing to speculate on potential avenues of treatment, and also at the very end, one area to be optimistic about. So you're definitely going to want to watch this to the end. And so this is the second time I had DrBeen on my podcast, and I can guarantee you it will not be the last. So thank you for watching. This is Dr. Josh. Peace and plants. DrBeen Syed is a physician, educator, and the founder of DrBeen Medical Lectures, with over 600,000 subscribers on YouTube and a growing following among clinicians and researchers. Dr. Syed is known for making complex immunology and pathology topics accessible and engaging. He's been one of the most diligent voices explaining spike protein pathology, both from infection and from vaccination. And today, he returns to go deeper on what the latest data reveals. Hi, everyone. It's Dr. Josh with Alzheimer's Breakthrough, and we're in for another treat here. Unbelievable. DrBeen has agreed to come back. We had over 100,000 views last time, and because there are so many questions and comments about spike protein and vaccines and things like that, he graciously agreed to come back. And let me start this way. Could one protein be the key to long COVID, to microclots, to the blood-brain barrier disruption, and even Alzheimer's? Today's returning guest, DrBeen, takes us beyond the surface into the persistence, pathology, and possible treatments for spike protein injury. DrBeen, thank you so much for coming back.
You're very welcome. Thank you very much for having me.
Awesome. So let's start here, because one of the things that's been very frustrating for me, and I'm sure for many of the listeners, is just trying to understand this whole area of persistence of the spike protein. Because if you read some papers, some articles will say, oh no, the spike protein will only stick around a couple days or maybe a week. And then there are other studies that we're seeing that, at least with people with long COVID, the spike protein can literally be around for years. So that's very confusing. And I know many of our listeners are confused, and, they're even talking about conspiracy, and I'm not thinking conspiracy. I think what's happening is people are different, but what do you think, DrBeen?
Absolutely. So let's first establish that the presence of spike protein after hundreds of days is possible or not. And yes, it is possible. And as you know, Josh, that I always look at various studies and provide you references for those as well. So I'm not just speaking out of thin air. So 3 ways to look at the persistence of spike protein, or 3 types of people. One are those who are immunosuppressed. For example, there was a patient in South Africa. She had uncontrolled HIV, and they saw that she developed SARS-CoV-2 infection, long COVID, but then she had about 300 or more days after, she still had spike protein present in her body, and that is understandable because she was immunosuppressed, and virus just kept sitting in there and kept making spike proteins. This is one type. The second is the patients who developed infection and then were not able to clear that infection, or were able to clear the infection, but somehow the spike is sitting somewhere in their body. Now, here is an interesting problem in this group, and actually you'll see that with the vaccine as well. That is it the virus persisting somewhere, or is it some other cells holding on to the spike protein? This is a mystery even to this date. You will see staunch advocates of viral persistence. They will bring up the topics about GAT, having the virus present in there for more than 59 days and so on. And they believe very strongly that the virus could be persisting. So again, now we are talking about a group that is apparently a healthy group with the correct immune system, not HIV or anti-cancer therapy patients. And somehow the spike protein is present in them as well. There is another group. For example, if you look at Dr. Bruce Patterson's work, and so people can, I have some studies open here as well. People can Google his work, Dr. Bruce Patterson and his team's work. They saw that after long COVID, the spike protein was present for hundreds of days in the monocyte cells. So what was it doing in the monocyte? The monocyte normally have a life of 3-4 days if they're not used up or become macrophages or active. Why are these monocytes containing these spike proteins? Why are they carrying forward and living for hundreds of days? So that is a mystery for them, and they have some speculations which I would discuss today. But it is seen that spike protein in long COVID patients has been there for hundreds of days, especially the monocytes that are on the sides of the blood vessels of the brain, so around the blood-brain barrier. That is why it is very common for spike patients, either from infection or vaccine, that they have brain fog and they have Alzheimer's acceleration and dementias and cognitive decline, fatigue, etc. So this is one group, the second group now, and that is apparently healthy group who is unable to clear out the spike protein. Is it being manufactured continuously, or is it just sticking in the cells and it is given over from one cell to the other, or one cell is just living for hundreds of days? But this is true that there are studies that are showing it is present. Now, the third group is the vaccinated individuals. And again, for all vaccinated, it is not necessary that they have spike present in their blood for a long time. In theory, the spike should enter after the messenger RNA or the other vaccine. The spike should be produced in the cells for 2 or 3 days at most, and then it should be cleared out. However, it is actually known now. I have this Yale Listen study preprint present here, where they said that in some of the post-vaccine patients, the spike was present 709 days after the event. That's
Wow.
a huge number of days, almost more than 2 years.
Right.
Why is that spike present in those cells or in that patient's body? And where is it coming from? There were some other studies that showed, these studies actually showed smaller duration, 90 days, 60 days, 60, 120 days, etc. They showed that the local lymph nodes ended up with the messenger RNA, and that messenger RNA got added to the cells present in the local lymph node, and now these lymph node cells are actually producing more and more spike protein.
Wow.
Why are these cells picking up those mRNAs? Why is the mRNA not going away? We know that synthetic mRNA is actually modified. For example, we have N1 pseudo-methyluridine, which is modified form of the uridine to allow that form to live longer in the spike protein, so that, or in the mRNA, so that the vaccine can stick in there for longer time to keep producing mRNA. Why is that mRNA persisting for a long time in the cells? Why is the local lymph node has become the factory to make spike protein? At least there are studies to show 90 days to 120 days in the local lymph nodes. Then the Yale Listen study, the preprint part for the vaccine, post-vaccination, shows up to 709 days. So the mystery for how this happens is there. But does it happen is clear now. There are many studies. So I hope that it was a long answer, but it made sense.
I think it makes sense. And then for some listeners here who are relatively new to this, when we talked about monocytes, they're just a type of white blood cell. And then with the last part of the answer, because that's what really confuses me, and I'm sure many other people, is when we're talking about the vaccine and the mRNA that's been modified to survive longer, it makes sense that it's with the methylpseudouridine that it survives longer in the body. But just to be clear, it's just an mRNA. It's not actually a virus. It's not something that at least was designed to last a long period of time. So it's fascinating in the study you just mentioned that it can survive for almost 2 years, not in the virus form, but in the mRNA form.
Correct. And I am just trying to quickly open this, for example, here in this study, let me first, so let me see what is the line number. It is line number 472. I'm going to go to the top of the study and read the title first. So the title is Immunological and Antigenic Signatures Associated with Chronic Illness After COVID-19 Vaccination. And this is Akiko Awasaki, Department of Immunology, Yale University School of Medicine, New Haven, Connecticut. So that is the study coming from. And let's now look at, it's a preprint yet. I doubt it would ever be printed because it is something to do with the vaccine. And I've seen vaccine studies not getting very good, chance to be published,
right,
because of the way our. So let's go to this line that I was going to read. So here on the line number 472, it writes, the manuscript says, most notably, we found elevated levels of spike, S1 and full length S, in circulation up to 709 days after vaccination among a subset with post-vaccine syndrome patients, even in those with no evidence of detectable SARS-CoV-2 infection.
But here's another key word you just wrote, a subset. So to
Correct.
be clear, you're not saying, the papers are not saying that everyone who's exposed to one of these vaccines is going to have persistence for years or a very long period of time. But there is a subset of people, and it would be great to know, and we were talking about this before. One of the things could be genetics. Not everyone has the same genetics, and it could be that people with mutations in receptors, etc., could either quickly remove the mRNA, the spike protein, or not.
Absolutely. Actually, the same study, which I just mentioned the title, this study is such a thorough study. I did a lecture about this study separately as well. It actually says that those patients who developed post-vaccine syndrome, they actually have a difference in their immune pattern or immune system's behavior compared to those who got vaccine and were healthier afterwards. And what they found was, it is such an interesting study. Actually, when I read this study, I was shocked, and I actually did the video saying this study shocked me. The shocking thing was, I had always thought that those who became vaccine injured, somehow their immune system becomes dysregulated and is unable to handle some of the metrics that happened or some of the inflammatory things, and it started attacking its own body through making antibodies or activating natural killer cells or macrophages or dendritic cells or cytotoxic cells, etc. That's what I thought. However, in this study, now keep an eye on the possibility of genetic changes or differences. In this study, they found that those who were post-vaccine and were injured, their immune system refused to downregulate in the presence of inflammation because of the spike, compared to those who got the vaccine and stayed healthy. What does this mean? Imagine you and I both get the vaccine, same vaccine. You got the vaccine, nothing happened. But what is happening inside your body is that the vaccine generated some sort of inflammatory state, activated some let's say cytotoxic T cells or dendritic cells or B cells, more antibodies on a longer term. And your body adjusted to that inflammatory state by reducing the activity of other cells to accommodate a little place for this extra inflammation that has appeared from somewhere. And imagine I got the vaccine and I got injured. What's happening in me is that the vaccine created inflammatory state in me as well. But my immune system, instead of trying to downregulate and accommodate and make room for vaccine generated extra inflammatory state, my system stayed normal. The shocking part was I was thinking that the patient's immune system actually becomes extra activated and causes more damage. But the problem was my immune system stayed normal, and now vaccine generated or induced further inflammatory state became it further slice up. And now you have inflammatory state. That has to be epigenetic or genetic. It could be environmental as well, or body habitus too, but I think this has to be genetic. And it is such a miss on our part as a country to say who are getting injured, what is their genetic makeup? Can we identify that makeup and then tell them beforehand that we don't think vaccine may be a good idea for you? We have never done that. But your suspicion is correct.
Wow. And it calls to effect that it would be nice to know, for example, if we could identify which genes would make you less or more susceptible with a vaccine, to actually do a genetic test on people beforehand and say, Dr. Bean, we'd love to give you this vaccine, but it looks like your genetics, your immune system is not going to be able to respond to the, or if it's going to respond too much and it's actually going to hurt you. So this would be like personalized vaccine development.
Absolutely. And I think that during COVID time, the whole world was in trouble. Almost everyone got the vaccine. There were trillions of dollars spent on this program. They could have used some dollars to actually do this research and help. Today, I was hearing that HHS is deciding to cancel or pause the mRNA vaccine projects of about $500 million at this time. Imagine this never had to happen. Imagine right from the mRNA time when people were receiving it, we were doing these studies to say what kind of a damage is it doing and why. And then we would have a small subset to say, you folks are going to become damaged with this. Your heart may be at risk, your brain may be at risk, your clotting system, your blood vascular system may be at risk. We think that you can get a N1 protein vaccine or maybe an adenovirus-based vaccine or something else. We just never did that. That's such a miss on our part.
Agreed. Especially when you're talking about vaccines, you're actually asking whoever's getting the vaccine, we're asking their immune system to do the work and produce the immunity. But if that person's immune system, for whatever reason, is not working well, then throwing a vaccine into it can be dangerous and counterproductive.
And I would speculate, or I will present, maybe their immune system is working well. And maybe others' immune system is responding to vaccines' inflammatory state and downregulating itself and kind of adjusting and accommodating. So who knows which one is working well? The problem is that there is a difference. For example, if I read the study, I want to, with your permission, I want to present some parts of it. So this same study, Akiko Iwasaki's study, and I'm just going to read some excerpts from it. They say, to explore potential pathobiological features associated with PVS, post-vaccine syndrome, we conducted a decentralized cross-sectional study involving 42 PVS participants and 22 healthy controls enrolled in Yale Listen study. Compared with controls, PVS participants exhibited differences in immune profiles, including, now what is the difference? Including reduced circulating memory and effector CD4 cells. So the first difference, that in those who became injured, they had fewer memory cells and effector or T helper cells, both type 1 and 2. So we have T helper 0, T helper 1, T helper 2, T helper 17, T reg. So T helper 1 and 2 both were reduced, memory cells were reduced, increase in tumor necrosis factor alpha CD8 T cells. So cytotoxic T cell, which were releasing tumor necrosis factor alpha, were increased. Then they say PVS participants also had lower anti-spike antibody titers, primarily maybe because of the vaccines. Then they say serological evidence of recent Epstein-Barr virus reactivation was observed more frequently in PVS participants. Further, individuals with PVS exhibited elevated levels of circulating spike protein compared to healthy controls. These are immune signature differences. And this is a preprint. And Akiko Awasaki and her team is an excellent set of researchers. It is a Yale Listen study which actually is looking at long COVID. And they did this favor to us being the medical community, to take this group and did this analysis. It is still a preprint. We are so cruel. We won't let any, and by we, I don't mean me, the healthcare system, it will not let any studies that can indicate a problem to become surfaced and published and recognized. Then when somebody is injured, they simply say, where is the evidence? We don't see any evidence. We don't see anything that shows causality. And that is such a circular state we are stuck in. Anyways, the topic started from, are there any differences in people who may become a subset? So yes, there are differences in them.
Awesome. And you used the word signature a little while ago. To my mind, when you said that, I thought, wow, just like people have completely different fingerprints, people have a different immunological signature or setup based on their kinetics. So, wow, that is fascinating. Okay, so
Absolutely.
If we could shift gears a little bit and focus on the brain, because we've been talking about spike protein. What about, because one of the articles you've put out there talks about the meninges and the spike protein
Yeah.
may go into
Yep.
the lining of the brain. What
Absolutely.
are your thoughts on that?
Yeah, so let's do this. I have it open here. It is printed in Cell, Host and Microbe. The printing date is December 11, 2024, so pretty fresh. And the title is Persistence of Spike Protein at the Skull Meninges Brain Axis May Contribute to the Neurological Sequelae of COVID-19. Now, the spike protein damage here will be the same with the vaccine as well. We have other studies, just like Yale study, that is showing that the vaccine-generated spike protein is sitting in the body 709 days after. So this idea that the vaccine spike protein will not do this is kind of a moot point, because that spike is also running around in the body and is detected. And that means it would go to blood-brain barrier, just like the virus as well. And I would present some studies that demonstrate it is not necessary to have the brain tissue problems only because of infection. Just the spike itself can cause the problems now that can come from the infection or from the vaccine. So here, persistence of spike protein at the skull meninges. So what they say here is, I'm just going to read some parts. Using optical clearing and imaging, we observed the accumulation of SARS-CoV-2 spike protein in the skull meninges brain axis of human COVID-19 patients, persisting long after viral clearance, meaning spike stays. Further biomarkers of neurodegeneration were elevated in the CSF from long COVID patients. So not only the spike was present, now it is causing inflammation and neurodegeneration. And the biomarkers, which I always get nervous sitting in front of you, a Harvard graduate with, brain diseases manager, and then I sit here and present to you various studies. So please, my apologies if I am
No.
overstepping.
You are a preeminent expert, so I love that you're covering this.
So I wanted to present this part. So they say that further biomarkers of neurodegenerations were elevated in the CSF in the long COVID patients. That means that the spike protein is present. It is causing inflammation. That inflammation is then causing neurodegeneration. And you and I both know one way of causing this kind of a damage is by activating glial cells. When the glial cells are activated, they're going to do synaptic pruning and they're going to start killing the neurons nearby. So that means some sort of inflammatory state is happening, which is sufficient to kill the neurons. That is just, it blows my mind. And then they continue and they say, and proteomic analysis of human skull, meninges, and brain samples revealed dysregulated inflammatory pathways and neurodegeneration associated changes. So they looked at the proteomics, that is the protein structures of the brain, and the skull and the meninges and the brain samples. And they saw that not only there are inflammatory pathways that are activated, but there are markers for the neuronal death as well. And we have seen that study. It was, I think it came out of Oxford, where they took the brain slices of the people who died. There are some folks in UK who have given their body for the scientific research. When they died, they looked at their brain and they took thin slices and kind of see the volume and do lots of other metrics on them. They took the brain volumes of the patients who had SARS-CoV-2 infection, and they say that even a mild infection caused up to 5% decrease in brain volume within a year. Now imagine somebody has spike protein in them for hundreds of days, which continues to be more than a year. How much brain volume damage will be happening? How many neurons are dying? How much trash is accumulating? It's a really bad situation for the brain. So back here, then they say, similar distribution patterns of the spike protein were observed in SARS-CoV-2 infected mice. Injection of spike protein alone was sufficient. Injection of spike protein alone, they're not talking about infection, sufficient to induce neuroinflammation, proteome changes in the skull meninges brain axis, anxiety-like behavior, exacerbate outcomes in mouse models of stroke and traumatic brain injury. Vaccination reduced. So what they're saying is, I want to explain this. Vaccination reduced but did not eliminate spike protein accumulation after infection in mice. So what are they talking about? They're saying if you give a vaccine to a mouse, then you infect them. Vaccination has some effect on reducing it, but it still is not sufficient to bring the spike levels down enough that they do not affect the brain. I am surprised that we ended up, I think naively, we ended up selecting spike protein for the vaccine. That is the main culprit for damage throughout the body. And we ended up selecting that. I think we should have selected some other protein.
I agree. I agree with you 100%.
Yeah. So this is a very interesting study as well, very thorough study, very exhaustive study that basically says spike, and they actually have a few videos as well. So once again, I'm going to read the title for your audience. Persistence of spike protein at the skull meninges brain axis may contribute to the neurological sequelae of COVID-19, December 11, 2024. There are a few videos where they actually show you, as they take slices of the brain and skulls and meninges, they actually show you, they've marked it with red, the presence of spike protein in bone marrow and on meninges and in the brain tissue.
Wow.
It's horrible.
And it sounds like it can spread easily. You know, I was thinking back to one of the ways that COVID would come in is through the cranial nerve number 1, the olfactory nerve, and then get back into the brain that way. But it sounds like because it can survive in all these different tissues, it just needs a nidus, it just needs a way, and once it's in there, it can explore and quickly spread.
Absolutely. So for those of us who know, like you, who know blood-brain barrier much better than I do, of course, the roof of the nose, the olfactory nerve, that is one pathway. However, there are studies that show that transcytosis, you are aware that endothelium in the brain is kind of barriered, right? So
Correct.
because it's the blood-brain barrier.
Normally, it should be barriered.
Correct. It is barriered, correct? And normally it should be barriered.
Yes.
And so things do not just willy-nilly end up in the brain tissue. And the problem with that is that as you seal the blood circulation away from the brain tissue, how are you going to get the nutrition into the brain? So for that, then those cells have specialized mechanisms. For example, there is transcytosis, there is receptor-assisted transcytosis, there are receptor-assisted movement of the molecules, there is aquaporin 4 in the glial cell, astrocyte food processes, etc. Interestingly, the transcytosis, you know that it is dependent a lot on omega-3 because omega-3 activates the proteins in the blood-brain barrier that causes the transcytosis to stay balanced and as needed by the brain. And interestingly, the spike protein and the inflammation caused by that causes transcytosis to become disrupted. So either it becomes so much that the cell is just a hollow ship now that you can just send whatever you like. The second thing is that the spike protein causes the plasma cells, the B cells, which are adaptive immune system cells. Activated B cells are called plasma cells. These plasma cells actually make antibodies against the blood-brain barrier. So you could sit out here and send antibodies. The spike could sit out here and send antibodies that would go and attack the blood-brain barrier and create the permeability and compromise the barrier to then have toxins getting into the tissue. And what happens inside the tissue? We have the monocytes that would become activated that are patrolling the boundaries, and we have the glial cells that would become activated. What are they going to do? They're going to start killing the neurons. That happens. So it is this study that I presented to you. It's very interesting because they have videos of macrophage activation, glial activation, spike accumulation in the meninges, brain marrow, and brain.
That's amazing. If you could talk a little bit more about the blood-brain barrier and pathways besides ACE2 for disruption of the blood-brain barrier with the endothelium, that would be amazing.
Absolutely. So there are two ways to look at that. So of course, one way that we are not going to look at is the spike binding to ACE protein and then causing endothelial activation and inflammation and compromise. So that's a known factor. Spike can cause damage to the blood-brain barrier or can cross blood-brain barrier in two further ways, big categories. One category is, as I just said, spike could be in the body. It could cause the adaptive immune system to become activated. It can cause the B cells to produce antibodies that are directed against the blood-brain barrier, which causes the blood-brain barrier to open up, and now the spike can get into the brain, right? The second thing is that there are multiple other proteins present on the endothelium, that is the inner surface of the blood vessels of the brain, present on the endothelium where spike can actually bind with them. It doesn't need ACE2. For example, alpha-5 beta-1 is another, or neuropilins. These are another. And there are tons of other proteins. I don't even remember all of them. Those proteins are present on the blood-brain barrier, and spike can bind with them and then become, this would be the receptor-mediated endocytosis. And so now the spike will be picked up, brought into the brain. Inside the brain, monocyte and glial cell will become very, very upset with the spike being present in the brain tissue. They are going to just become so angry. They're going to think there is something wrong in the brain tissue. They're going to start pruning the synaptic junctions, and they would kind of disentangle, disconnect neurons from each other. The result of that is that we'll have severe brain fog, we'll have issues with memory, we'll have cognitive decline. If somebody is already dementic, they would have accelerated dementia, Alzheimer's would become accelerated. All brain conditions can become intensified. I was actually reading some studies, even epileptic patients would have exaggeration and exacerbation of epileptic situation. So not only now that the glial cell are activated and causing synaptic pruning, they would actually help kill the neurons as well. And then another very interesting thing, and my once again apologies, speaking about brain in your presence is kind of showing a light bulb to sun, but hippocampus, that hippocampus has it is connected with the memory. It's a gateway for the memories, right? And cognition. So whenever people who are listening to this right now, what's happening is this information is passing through their hippocampus, and then hippocampus is helping make them into memories. If some part of it will be remembered, hippocampus is involved in that. Hippocampus has stem cells. Stem cells, by definitions, are the cell that can make their own type and they can make new types. There are stem cells in the hippocampus that would help produce new neurons. Hippocampus produces 500 new neurons every day in us. By the age of 50, 51, the whole hippocampus has become replaced because of the new neurons that are produced and the old neurons that are removed. When there is inflammation because of spike protein, it causes the stem cells in the hippocampus to die. So now imagine, in general, normal activity of the brain, we need 500 neurons to be killed and 500 neurons to be produced in hippocampus to fade out the older memories and to make newer memories. And here you come with a spike protein and kill the stem cells that are supposed to do this. Now what's happening? Neurons are dying, but new neurons are not forming. Old memories are fading away as they should, but new memories are not being built as efficiently as we expect them to be. And this is what your patients are going to say. Hey, I have brain fog. I don't remember things. I stand in a room. I do not know why I was here, right? I am becoming more forgetful. I cannot do planning. I was trying to look at something and I was trying to solve some problem and I can't do it. I cannot manage an Excel sheet. What's happening is they are getting inflammation in the brain. I'll stop here. Otherwise, I'll keep talking about this forever.
Right. But what I'm thinking is it's not just the hippocampus. I mean, you're talking about brain fog and new memories. That's normally 4-second memory, which is in the frontal lobe, which is not the hippocampus. So unfortunately, the spike protein we found is not just affecting one small part of the brain.
It's the whole brain.
Yeah.
I just gave an example to kind of connect it to closely to us. It is the whole brain that is affected.
Bingo. So tell us more about, because when you were talking about endothelial activation and disruption, there's also this concept of microclots that can turn into macro clots or larger clots. And obviously we see this in the whole body, not just in the brain, but that's obviously where I like to focus. But I think you found another article specifically about the endothelial activation and microclots. What did that study show?
Absolutely. So a couple of studies here that are interesting. One, I want to go back for a second to microglia, and I wanted to sensitize your audience to a very scary but interesting thing. If you ever Google a term, fiery death of cells, I think it was a news a few years ago, I think after the COVID-19 or something, where they said that cells develop a fiery death. And the whole country was, wow, we have fiery death. What is that? So microglia that we were talking about, the glial cell that are present in the brain, these are part of the defense system of the brain. When they become activated with this spike protein, they actually can cause fiery death. And what does that mean? What happens is such cells, before they die, they tend to die. They decide to die. But before they die, inside of them, they start producing a bunch of toxins or inflammatory cytokines. This happens because of NLRP3 activation onto these cells. And now the cell is destined to die, but it just keeps packing a lot of cytokine material in it, inflammatory toxins in it. And when it dies, it opens up with all of those chemicals released everywhere in the tissue, causing inflammation, widespread inflammation and tissue damage. Immune system become activated and start killing those things which are showing those damaging cytokines. So that is called a inflammasome-based inflammation. So the spike protein present in the brain also causes NLRP3-based activation of microglia, which then cause this inflammation and tissue damage. Now going to the clotting. So I'm going to quickly go over some of the studies that are related to this so I can kind of read them to you, or the titles. So just give me 1 second.
Creatine is back on people's minds. You've probably heard, not just because it helps you build and maintain lean muscle, but because it may also be beneficial for the brain. That's why I'm so glad my friends over at Organifi have created this awesome new product, Creatine Cherry Chews. People have been taking creatine for a long time now, but it's mostly been professional bodybuilders and elite biohackers. Now, Organifi has transformed it into something that everyone can take every day and really should. Organifi always goes the extra mile in finding the finest ingredients on the planet, and the same goes here. They've sourced the best, most bioavailable creatine monohydrate, which has been shown to help you push yourself further with less effort. Studies show that it also helps support cognitive function and brain health, which makes taking creatine a great choice for everyone. Plus, Organifi has added tart cherry, which is actually a superfood loaded with antioxidants and helps your body recover quicker. It's really the perfect combination. These two ingredients come together in a seriously yummy, soft chew gummy, and now it's convenient to take your creatine anywhere you go. You might be thinking, but I've heard that gummies are just loaded with sugar and rarely contain an effective dose of the intended ingredients. And if we were talking about one of the competitors out there, that may very well be true. But with Organifi, you get an effective 3 grams per serving, which you can adjust to your preference, and you're not getting any added junk like artificial sweeteners, flavors, or tons of sugar. I know you'll love the Creatine Cherry Chews, and I know you'll love this too. I'm going to save you some money. Because you're listening to this right now, you'll save an extra 20% off their already low price. Just go over to Organifi.com/Dr. Josh. That's ORGANIFI.com/DRJOSH and grab your Creatine Cherry Chews. Remember to use my discount code Dr. Josh at checkout to save an extra 20%. Again, that's Organifi.com/Dr. Josh and use code Dr. Josh at checkout to save 20%. And while you're looking in terms of the fiery death is actually can sometimes be adaptive if in the death they're trying to kill infection. So it's not, there's actually a reason why sometimes you want cells to do this as they're dying.
Correct. Okay, so a few studies around the clotting aspect, and maybe a doctor to look up as well. There's a doctor, her name is Dr. Rasia Pretorius, if I'm pronouncing it correctly. She is from South Africa. Very recently, she was here with Harvard. She's been working with Harvard to do some more research about the clotting aspect of the spike protein, regardless of the COVID or the vaccine. She had this hypothesis that somehow the clots that are produced because of the spike protein are less dissolvable, less dilutable, less degradable, less breakable. Normally what happens is, in all of our body, there are tiny microclots that are formed because the blood cells are and platelets are kind of bopping around in the cell. There are branches to the blood vessels, and the running RBCs and platelets hit the branching area and they can become entangled there. Then in our legs, we have blood that is slowly moving upwards, and in those veins there are valves, and at the corners of the valve there is hypoxic conditions, and the clotting can form there, meaning there are many reasons that we may develop clots in our body normally. Sure. And those clots are then broken down. And when they are broken down, we can see D-dimers present in the body, and there is a specific levels of that and so on. If we make even more clots, we would try to break them down as well. Racia Pretorius' point of view was that when there are clots formed by the spike protein, the clot breaking enzymes are trapped into the clot itself so much that they cannot become activated. Normally, clot has a clot breaking enzyme trapped in it as well. They become activated at some point and start dissolving and degrading the clot. But her point was that they are not able to become activated. So now we have a clot. It is stuck on the blood vessel surface, but it is not going away. It is not washing away. It is not degrading. So these are persistent clots. And the more they are, the smaller blood vessels, the more impact to the blood circulation. So keep Racia Pretorius and her work in mind. And I'm going to present some more studies here. So there is a study here. This is January 2024. Study title is SARS-CoV-2 and the spike protein in endotheliopathy. So you would read new diseases like spikiopathies is a term that has been used. I've been using that as well. More researchers have been using it. Endotheliopathy is endothelial damage. So here they're saying,
and the endothelium is just the lining inside blood vessels.
Correct. Inner surface of the blood vessel, including the blood-brain barrier. So here they are saying endothelial cells are located at the interface between blood and interstitial tissue and are a key player in vascular homeostasis or health of the blood vessels. Endothelial cells are in a dynamic equilibrium with their environment and concomitantly constitute a source, barrier, and target of defensive mediators. A large body of evidence indicates that endothelial cell dysfunction across different organs is a major dictator of the severity of SARS-CoV-2 infection. I would replace here, not with, not changing the manuscript, but the spike protein. And what they say is, more recently, a rapidly growing number of reports have indicated that SARS-CoV-2 can interact directly with endothelial cells through the spike protein, leading to multiple instances of endothelial dysfunction. So there is another study by Dr. Bruce Patterson about the endothelial damage and clotting as well. So let me explain another mechanism. So this is one mechanism. They're saying that the spike protein interacts with the endothelium. Dr. Bruce Patterson and his team found that what happens is that when spike protein interacts with the endothelium of the blood vessel, it causes SCD40L or CD40 ligand to become activated. It's a protein that would become activated on the surface of a cell. It causes platelets to become activated. Activated platelet actually causes CD40L to be activated, so there is a cycle there. In addition to that, as CD40L, platelets that become activated because the endothelium had become activated. So let me back up for a second for our audience here. Imagine a pipe. Inside that pipe, there are platelets and red blood cells and other things, the blood. And as some spike protein kind of knocks onto the endothelial cells, they become inflamed. When they become inflamed, they become activated. When they become activated, the platelets that are running around in the blood vessel, their job, the platelets' job, is to see if endothelium is activated. Because activated endothelium is a sign of injury. And platelets' job is to make a clot at the area of injury to prevent bleeding. So when the endothelium is activated, platelet becomes activated. When platelet becomes activated, they want more platelets to be activated. They want more endothelium to be activated. They want to make a clot because they think there is bleeding that is happening. So what they do is they release even more as CD40L, plus they release VEGF, vascular endothelial growth factor. Activated endothelium also releases VEGF, or vascular endothelial growth factor. The result of that is that the endothelium is activated, endothelium is inflamed, platelets are activated. Activated platelets are now binding with each other, creating clots. Imagine those clots being formed in the blood-brain barrier area. Do you think that the brain is going to get blood circulation in a normal way? No. Right? So now we have the smallest part of the brain, for example, internal capsule of the brain, has very tiny, delicate blood vessels. You start putting clots in there, this patient is in trouble. Right? So now what happens is that as clotting starts happening, and as the endothelium becomes activated, the endothelium does another bad thing. And that bad thing is that it starts producing cell adhesion molecules. Why is it doing that? Look, nobody knows the endothelium or platelets that we have spike protein around. When endothelium got a signal by the spike protein, endothelium thought there is a damage somewhere. When the platelet is activated, platelet thinks there is a damage somewhere. So everybody is now busy creating clots, while the culprit is just a simple spike protein sitting somewhere laughing that, hey man, look what I did. Now, when the endothelium and the platelets, they are activated, what they would do is they would cause even more activation of endothelium, which would cause smooth muscles to start becoming contracted. So blood vessel narrows down. The nitric oxide production is reduced inside the smooth endothelium that goes to smooth muscle and causes the diameter to increase. So now blood vessel is constricted, has clotting propensity. Superoxide dismutase, this is another enzyme that is present in the endothelium of the blood-brain barrier and all the blood vessels, and it helps reduce inflammation. Superoxide dismutase is reduced. Reactive oxygen species are increased. This is a hugely important clotting and inflamed blood vascular system, a system with clotting. Now, interestingly, these clots are not even erased or degraded easily. And this is why you would see some of the doctors talking about triple therapy or various kind of anti-clotting and blood thinners to be given. And some patients feel better with that. So anyways, clotting is a very important topic, and these clots seem to be a little more sticky than compared to normal clots.
Fascinating, especially the fact that these are different types of clots that are much more difficult for the body to get rid of, to lyse. And I can see how these microclots turn into much bigger clots. And for example, in some of my patients who had lung problems, with you can see huge clots in their lungs, as an example.
Right. Yeah. Wow. So because these are a bit more sticky clots.
Wow. So the next thing I wanted to ask you about was, there's an article about comparing vaccine-derived and infection-derived spike protein in terms of the biodistribution, where we see it. What are your thoughts on that paper?
Understood. So I think a part of this we have already discussed, that in the beginning there was a thought that only the infection-derived spike protein can end up in the brain because the virus goes there, then virus binds, and the virus is replicating and increasing in number, and that is why that spike protein is in the brain. However, now we are seeing, as I talked about, that Yale study that said 709 days after the vaccination, without any understanding or knowledge of the infection or proof of the infection, the patient only had the vaccine. 709 days after, they have their spike protein present in their circulation. So this idea that used to be prevalent in the beginning, that we would have spike protein coming definitely from the virus, and the vaccine-based spike protein will actually not be running around in the body and not biodistribute in the tissues and would just go away after a few days. That idea is now totally incorrect. And there is one study here, the Yale study, 709 plus days. Then Dr. Bruce Patterson's study, he just did this study. So he had done a study earlier where he showed spike protein in long COVID patients even after 1 year. Then he and his team did one more study that actually came out very recently. So let me actually look at the studies, see the exact date when it came out. I had it open here. 1 second. Okay, so this study, it is lost in my treasure trove of studies. But if you just look for here. Detection of S1 spike protein in CD16 plus monocytes up to 245 days in SARS-CoV-2 negative post-COVID-19 vaccine syndrome patients. SARS-CoV-2 negative and vaccine positive patients S1 in their monocytes 245 days later. Almost a year. And they actually have a theory. I promised you in the beginning of the talk that I'll explain why did they think it is happening. They think that in these patients, first is they have an observation. Their observation is that these patients have less tumor necrosis factor alpha and more interleukin 4. Interleukin 4 primarily by some unknown cells plus from the T helper 2 cells as well. Remember, T helper 2 cells produce interleukin 4 and 5 and interleukin 10, and 4 and 5 help with the maturity of the B cells and class switching, etc. They see that in these patients, interleukin 4 is more and tumor necrosis factor alpha is less. And what they found was that in a normal monocyte, it is the tumor necrosis factor alpha that triggers the death of monocyte. Right? So the monocyte has to live only for a few days, maybe a week. It is born in the bone marrow, it is circulating in the blood circulation. If it didn't get converted into some resident cell for a long time, it would die. If it is not used up, it dies. When it dies, it happens within 3-4 days, a week later maybe. And one trigger for it to die is tumor necrosis factor alpha. And Bruce Patterson and his team saw that in these patients of the vaccine I'm talking about, they have more interleukin 4, less tumor necrosis factor alpha. Interleukin 4 actually causes the reduction of tumor necrosis factor alpha. And less in tumor necrosis factor alpha means that the monocyte will not receive the death signal. And that monocyte can continue to live for a long time. So what is the solution then? The solution is that you reduce interleukin 4 and increase tumor necrosis factor alpha, which then gives a message to monocytes to say, hey man, you guys can go away, and then they die and they take the spike protein with it. So that is a very interesting state. And what did they find? They actually, if you look at all of their studies, they always talk about 2 drugs to be used. One is statin, because the statin works with the fractalkines. Fractalkine is another way where the platelet activation and platelet interaction with the endothelium happens. Fractalkine is a tree-like little molecule, microscopic, that grows on the endothelial surfaces. And then that allows the platelets to activate with it and cause clotting. So fractalkine can be disabled by statins. And then the CCR5, which is another molecule that is important for this, what was the second mechanism? Was monocyte.
Right.
So for the monocyte, you can give CCR5 blockers that would then reduce monocyte activation. And for them, it is Morovirog. And now I have heard many patients who say Morovirog doesn't work. Many patients say it works for us. But their intervention suggestion is for this kind of a situation with clotting and monocyte activation and brain fog is statin plus Morovirog.
Excellent. And it'll be interesting. Hopefully they're looking at the genetics, because it could be there's a genetic difference between the responders and the non-responders to these different pharma approaches. Shifting gears a little bit here, explain, if you could, the overlap between the persistent spike proteomics or protein expression with some of the Alzheimer's markers.
That is a beautiful question. So first, let's set the stage to say can the presence of spike protein trigger or induce dementia or accelerate that? So I'm going to just read a quick study here. And then in that study, we'll see how do they feel that this is connected to the proteomics. So this is a study, it is in ACS publications, and the study title is Spike Protein Fragments Promote Alzheimer's Amyloidogenesis. And this is August 16, 2023. The study has an abstract, which I'm going to leave most of it. I'm going to read a part of it, which is very interesting. Here we first identified the amyloidogenic potential, that is amyloid production potential, which can give rise to dementia. Amyloidogenic potential of HGVV FLHVTYV. So people would say, what happened to Mobeen?
That sounds like a crazy gene or something.
This is a gene for spike protein, and the protein area that it makes is actually something that can interact with the amyloidogenic proteins. So they say that potential amyloidogenic potential of this area, a short fragment of the spike protein of SARS-CoV-2 coronavirus, the peptide fragment, this short fragment, protein fragment, was found to be toxic and displayed a high binding property for the amyloidogenic segments of A beta, thereby promoting the aggregation and toxicity of the peptide in vitro and in silico, while retarding the hatching and survival of zebrafish embryos upon exposure.
Wow.
So it has a fragment, spike protein, that can bind with A beta and make it amyloidogenic. That is, then it gets cut in the wrong places and the plaques are formed. So spike protein directly helps produce amyloids.
Wow. So that means that some of the patients I'm seeing with Alzheimer's who have high amounts of amyloid beta in their brain, one of the causes could be this small fragment of spike protein you mentioned.
Correct. And they say our study implicated SARS-CoV-2 viral infection as a potential contributor to Alzheimer's disease pathogenesis, a little explored area in our quest for understanding and overcoming long COVID. So once again, it can be extrapolated to spike protein from the vaccine as well. But they actually found the exact area of the spike protein that interacts with A beta to then cause amyloidogenesis, to then accelerate Alzheimer's disease or induce Alzheimer's disease.
Fascinating. And so this could also explain why we're seeing more and more cases of Alzheimer's and other amyloid-related brain diseases.
Yeah.
Just
So
because,
sadly, either someone got infection, or they got vaccine, or they got infection and vaccine, or vaccine and infection. But somehow, all of us, majority of us, are exposed to spike. And we are talking about spike doing this. So I think we have scarred, either knowingly or unknowingly. The virus infection is an unknown thing. I mean, I do not know how much control do we have. Vaccine was a more knowing thing to be done. I don't think they knew that this would happen, but they never let this kind of education and science to become more prevalent either. So there was I believe, a deliberate bad aspect of decision making. But anyways, we're seeing here, and almost all of the world has become exposed to these things. Very few are protected. So we are going to see, I think, in the near future, we're going to see a number of cardiovascular diseases in unexpected places, meaning unexpected people, younger people, healthier people. We'll see a number of clotting diseases that would become prevalent. We'll see more autoimmune diseases in women who already are prone to autoimmune diseases. You would see them having more autoimmune and more intense autoimmune diseases. We would see more brain fog and more cognitive decline and more dementia-like patients. You would see more diabetic patients because the pancreatic state of inflammation would make people prone towards diabetes. Plus, generalized body inflammation would cause insulin resistance because the cells are not able to function correctly, which would cause hyperinsulinemia, that would cause resistance to insulin, and that would cause diabetes. So you would see a lot of fatty liver disease. You would see a lot of muscular diseases. These diseases will ramp up. Now, people always bug me at this point to kind of say, why aren't you talking about cancers? Cancers would increase too. I do not have enough good studies to be able to connect this to cancers. I can say in general that anybody who has a higher state of inflammation and cell division, they are at risk of cancers. But I haven't yet seen those direct linking good studies. That is why I keep my mouth shut about cancers.
Wow. So that's a call for people like us and others who are watching this in the medical field to come up with other approaches to try to reverse some of these problems, because from my experience and from what you're describing, the numbers are just going up and up, or we're seeing it at a younger age. I'm seeing dementia at younger ages and other chronic diseases at younger ages, and it now makes sense why we're starting to see that.
Yeah, this whole generation of billions of people, we are scarred at this time.
Wow. So shifting gears, let's talk about how the body can get rid of the spike protein, how it can clear the spike protein. What mechanisms are there, whether it's microglia from the immune system or the lymphatic drainage system? What are your thoughts there?
Very good question, and very less science available. However, I would, for your audience, number one, I would ask them to be protected from snake oil salesmen. There are very common uh, messaging that I see, narrative that I see, detoxification from spike. My product can dissolve the spike. We will remove spike from your body within 10 days. Or some of the doctors, with their possible solutions, they make their patients an ATM machine. And they say, well, you're going to pay us $600 monthly, and we are going to send you these herbs and supplements and blah. And they would help you with this. And you just keep giving us the money and we'll keep sending you this. So there is a lot of incorrect practice, malpractice that is happening. And lots of people are desperate and they are losing money. And I can understand desperate. During a couple of times when I became infected with COVID, and maybe even after the vaccine, I developed a lot of muscle fatigue. I would come upstairs and I would just be like tons of weight is being lifted by my legs. And I felt I am gone. My life as I know it, healthy life, is gone. I can't be like this forever. And so I can imagine people who are more damaged, they are desperate. And so they're looking at anything and everyone to help them. So just be protective of yourself. I think go to Dr. Josh, and he's a graduate. He has well studied. He understands these things. Maybe he can help better than someone who says, I'll give you this magic soup and you'll become better.
Thanks.
Having said that,
Yeah.
there are certain principles of management that would apply. One principle is control the inflammation. Now, controlling inflammation does not fix the problem, but it fixes or reduces the damage done because of the problem. So inflammatory control is very important. In that area, omega-3 is very important. Reduced omega-6 is very important. Omega-3, especially Dr. Josh's patients are more on the brain diseases side and want to understand that more. Omega-3 is especially good for the blood-brain barrier because omega-3 works with a protein in the blood-brain barrier that helps with the health of transcytosis. Transcytosis is a transport mechanism to take things in the blood and bring them to the brain. And if omega-3 is not present or is less in amount in the brain tissue, blood vessels, that would cause the transcytosis to go rampant, and that would cause leaky brain or compromised blood-brain barrier. So omega-3 is very important, less omega-6. One can take other anti-inflammatories. For example, curcumin is a very good anti-inflammatory. At the end of the day, you have to decide a number of them that you can take, one or two, not all of the ones that I'm speaking about. Then there is another set after anti-inflammatory. Vitamin C can be part of that as well. Then if you go to another set, and that is around the clotting. The clotting has a specific problem. These are more sticky clots. So of course that there are things like nattokinase and lumbrokinase. In addition to that, there is this NSE. And why do I say NSE? NSE, or N-acetylcysteine, is a product that helps. It used to be for cough, because what it does is it breaks disulfide bonds. So whenever there are things that are gelling together and becoming a firm gel, usually there are disulfide bonds that are developing there. And NSE can break those bonds. And this is why it used to be used as an expectorant, because it would break the bonds in the developing phlegm and kind of liquefy that and help clean the lungs.
I can say it still is used that way, but yes.
Yeah. Thank you. So NSE is another very important one that can be used to help reduce the firmness of the clot and kind of make it more liquidy and try to break it down. Similarly, other inflammatory tissues, especially in the brain where the extracellular matrix is increasing, ECM is more collagen and more matrix or the fluids that are accumulating as a result of inflammation. NSE would actually help keep them dissolved and thin and allow them to be washed away. Then resveratrol is very good for binding with the spike protein. Then there is another thing that we did not discuss today. Maybe we can do a separate discussion. That is mitochondria. Mitochondrial damage occurs with all of this problem. So for mitochondria, methylene blue, very low dose. And there is methylene blue that is industrial for the clothes or the tissue staining. Not that one, but there is therapeutic or pharmacological methylene blue as well. And just 1 gram or 2, very tiny amount. It is used for carbon monoxide poisoning, but a tiny amount can be used on daily basis. It can make your mouth a little blue, but methylene blue is an electron donor and electron receiver. It kickstarts the mitochondria that are not working correctly. So that is a very interesting product to be used for mitochondrial health. Near infrared light. It can penetrate up to 4 inches in our body. It is very good for the mitochondrial health. Don't use it for more than 10 to 15 minutes at one part of your body. You could use it for 10-15 minutes in the front, then use it 10-15 minutes in the back, but not more than 10-15 minutes in one area. It starts becoming toxic after that. But lesser than 10 minutes is not useful. More than 15 minutes is less useful. 10 to 15 is a good window. Near infrared light. What else? For clotting, lumbrokinase, nattokinase, or other blood thinners, do talk with your doctor, because when it comes to blood thinners, you are at risk of bleeding, and that can cause strokes and heart attacks and other problems. So that should be done with your doctor's help. Now, there are more ways to help with the outcome of the pathology and not the spike protein itself. For example, there is IVIG that helps bind with the antibodies that are being produced. There is plasmapheresis that Razia Pretorius is very fond of that, and she has lots of centers for that as well. They clean the blood plasma, and that helps for a few months because spike is still being produced. Resveratrol actually directly binds with the spike. Now, to get these monocytes to die away, interleukin-4 blockers and the tumor necrosis factor alpha producers are more interesting. So Maraviroc is a very good thing to try. It's not necessary that you will become better, but it is possible that you will. And if that works for you, there are many patients who said we were bedridden and now we are up. So if that subset is something that you are part of that, then it would really help there. statins, as I said before, they help disentangle the platelets from the blood vessels and reduce the clotting as well.
They do, but unfortunately, they can also cause damage to the
Absolutely.
mitochondria,
Yes.
deficiency with CoQ10. I'll just put a
Yes.
disclaimer with all these. I love this list, but I would say
Yeah.
with all of these, definitely consult with your own physician because
Absolutely.
so for
Absolutely.
example, with the methylene blue, not only does it help with potentially clotting, it also happens to be an antibiotic. And so if you do things too quickly, you can cause a Herxheimer reaction. It also is one of the original psychiatric medications. So then that's a whole other discussion, no question.
Correct. So all of these drugs and interventions with your doctor's help, of course, I'm not sitting here and giving a medical advice, but if you wanted to have a list of possible potential therapies, these are the lists, and then you can sit down with your doctor. These are primary things. The list can keep going. I've been teaching it for 5 years, but this is the main list.
Awesome. So, and I would also put a proviso. One thing that I'm finding helpful is actually doing genetic testing on my patients before selecting and figuring out which specific genes we're trying to fix using resveratrol or these other options. So, shifting gears a little bit here, tell us more about the pattern recognition receptors, the TLR toll-like receptors that are actually activated by the spike protein.
Correct. So as I mentioned before as well, the microglia. So let me back up for a second. Any foreign agent, spike protein or something else, will trigger pattern recognition receptors on our cells that bear those receptors. So for example, some of our cells like macrophages, dendritic cells, natural killer cells, neutrophils, some other cells, they have little receptors on them, outward and inward, to sample their environment. These are called pattern recognition receptors. And then there are subclasses of those. And one subclass is TLR. Similarly, there are NLRP. Then TLR itself is of multiple type, TLR 1, 2, 3, 4, etc. Some of those are on the outside, some of them are inside. The cells are actually continuously sampling their internal environment as well to see if everything is okay. The microglia and NLRP receptor, which is a type of a pattern recognition receptor, when that sees the spike protein, it becomes activated to make inflammasome and kind of burst open the mitochondria. In the other cells, for example, dendritic cell and the macrophages, when they see the spike protein through their PRR4s or PRRs like TLR4, they would actually pick up the spike protein or the messenger RNA related to it and bring it to the lymph node. One of the reasons, Josh, this may be interesting, Dr. Josh, one of the interesting things that they found as part of their speculation for the spike protein after messenger RNA being formed in the lymph node, they are thinking that what happens is that the dendritic cells here in the area of injection pull those sick cells and the material coming out of them and bring that material to the lymph node, which is their job. In that process, they bring messenger RNA here as well, which gets trapped into dividing cells, and now it is just dividing with them and it is present in them. Another possibility is exosomes are leaking with the messenger RNA from here, which comes in those areas. So when we have those foreign material being produced, macrophages, dendritic cells, microglial cells in the brain, they'll become activated, and then the inflammation and the damage, as we've been discussing, would happen.
Wow. So, and you may have touched on some of these already, but what are some of the therapeutic candidates or interventional studies, like, maybe coming up with antagonists against these TLR toll-like receptors or anticoagulants? What are your thoughts there?
So there is not much for these studies. So if I tell you official big studies that we have seen so far, that these are corticosteroids that give corticosteroid and that would help the patient, and that is good. The other studies, for example, looking at these groupings for bleeding disorders or clotting disorder and combining that with the blood thinners, there have been very few studies. So from an emerging intervention point of view, the only studies that I'm seeing are some things related to statins, Maraviroc, natokinase, heparin studies, IVIG studies, plasmapheresis studies, and anti-inflammatories in general. I haven't seen official big studies for example, resveratrol, NAC, curcumin, omega-3, amla green, or ashwagandha, or lumbrokinase. So some studies are present, but they are really limited.
Right. And of course, with these studies can be very expensive to conduct. And if funding is tight, it's hard to justify. I mean, it's one thing if you hold a patent on a medication that you can profit from in the future years, but if it's something over the counter like ashwagandha, there's unfortunately not a strong financial incentive to do the study.
Correct.
Wow. We've talked about some pretty involved and frankly some of it's pretty depressing. Is there anything we can be optimistic about with all this?
So the only thing that I find it interesting is that in a subset, so there is a subset that becomes vaccine injured or long COVID. Within that subset, there are multiple types further. But I've seen that in a subset, their symptoms continue to reduce over time. So that is a subset. So there is a hope for one group of people. And if I then juxtapose that with there is another subset where if you do not intervene, if you do not step in and try to break that cycle, they just continue to become deteriorated. More and more brain fog, more and more Alzheimer or dementia situation, more and more memory problems, more and more clotting issues, more and more muscle fatigues and cardiovascular issues. So there is a subset that just keeps deteriorating until you step in and try to stall it. And there is a subset that can actually recover. So the only good news, I think, is that there is a subset that recovers. The second good news is those who are suffering, a number of them can actually be feeling better with these intervention. And the third good news is that there are a lot more studies that are coming out. I hope that, and I sincerely hope that RFK Jr., it's not necessary that I agree with all of his science, but if RFK Jr. stays in his position for some more time, I wish he does, there may be more vaccine-related science that would be allowed to appear. He says something about science which you don't agree, fine. However, and by you I mean someone, however, even if we allow the good researchers to look into the vaccine or to look at the long COVID and to come back and say, here are the genetic changes, comorbidities, body habitus differences, environmental differences that can increase the risk, then I think we'll have lots of benefit. There is a feeling in me that one reason that these studies are not allowed is because if these studies happen and there is a causal relationship of a damage to the vaccine, then the insurance companies or the government, somebody has to compensate for that. And the more time that is passed with saying there is no causal relationship, it's just association, temporal association, maybe vaccine, maybe something else, we don't know, that would keep the government protected from paying out. But I think that is a horrible thing to the vaccine patients.
Oh, yeah. So I've asked you a lot of questions. Was there any question I should have asked you, but didn't?
No, I think that you have actually asked a number of very good questions. At some point when you would like, we can do another discussion about mitochondria. So I believe that mitochondrial health and mitochondrial damage because of spike protein has been a topic that you and I have not went into depth as we did in this one and the last one. And mitochondrial health is very important for brain fog and dementia and memory as well. So maybe at another time, you and I can have another discussion only related to spike protein and mitochondria and brain health. And that would be an interesting topic.
I love that. I love that idea. So let's definitely plan for that. And so Dr. Bean, if people want to find you online, social media, how can people locate you?
So Dr. Bean Medical Lectures. One is simply drbeen.com, DrBeen.com. So that is my site where I have medical lectures, which I sell them, and there are CMEs with them. If you wanted to just see my free lectures, then you can go to YouTube and look for Dr. Bean Medical Lectures. There are two channels actually. I have Dr. Bean Medical Education, which is a newer channel, which is only medical education. And then there is Dr. Bean Medical Lectures, which is an older channel with a bigger audience, where I have been discussing vaccines and COVID and long COVID and medical. And all. So it's more of a mixture with the contemporary sciences. So that is where you can find me.
Wow. Dr. Bean, thank you as usual for your amazing expertise, your willingness to dive deep and study this whole field, and then most importantly, to share it with my audience. I salute you. You've identified an area of huge concern, but also an area with potential for improvement of Alzheimer's and other chronic conditions. So thank you so much, Dr. Bean.
You're very welcome, and I hope this can be useful for some of the patients and people who listen to this. Thank you for having me.
Awesome. Peace and plants.